Thanks to everyone, including the participants, the attendees, great questions. And special thanks to Roger as well for convening us, but also to our MHRP colleagues, Julie and Sandy, I think, who really provided a lot of impetus in pulling this together. How to remark on this is impossible a task, really, because we've seen such an incredible breadth of ideas and background data. And for me, if nothing else, it's an opportunity to reflect upon where we are today in this HIV pandemic as we've lived it over the past 30 years. And notably, in the past year, we've been so impressed by the vaccinology that is getting us out of this pandemic and the urgent need, of course, to apply that to regions of the world which desperately need these vaccines. But it's remarkable also to think about how that development of the COVID vaccines has built on literally centuries of progress. And that what we've seen in the past year is kind of this typical vaccine development for a pathogen that's fairly easy to find a vaccine for. I mean, I think all of us would agree that that's the case. However, as Guido called it, this little sucker, this HIV virus is very, very different. And what we've seen today is notably many of the challenges that we've come up against in trying to find an effective vaccine. If we reflect upon the astonishing success in the past year and think about if rather than greater than 90% efficacy against moderate severe disease, we saw moderate efficacy. If we saw 50% efficacy, how much of the effort that we're focusing on here with HIV would be highlighted, would be invested in, would be further pursued. And for me, it just is a remarkable opportunity to again remind us for the need of humility in pursuing these scientific endeavors, and the recognition that we don't know yet really how to advance an HIV vaccine. Now, hopefully in the coming months, and Daniel and the 705 and the Mosaic, the Embokodo and Mosaico teams will provide us some signal in which we can pursue further. But I think we're also expecting that to be moderate at best. And so everything that's been discussed today, from setting the stage with Georgia and Dan, to the behavioral assessments, the mucosal activation, these innate immune analyses and FC receptors, and the gene signature, Rosmi, is all fascinating. If we need to apply these efforts and investigations to an HIV vaccine, there's no question in my mind that with the humility that we must embrace and acknowledging that we don't know the answer, we need to investigate these as deeply and as profoundly as we possibly can. So that's all I have to say, other than a tremendous thanks to everyone, and in particular, the participants, the people out there in the world who volunteer for these studies, providing blood, sweat, tears, mucosal samples, to advance our efforts in combating this very serious pandemic. And thank you all.
Thank you, Jim. Great words. I'm going to close this meeting. I would like to thank all those who attended, all presenters, my co-host and co-organizer, the MHRP and HVTN. As Jim noted, we have heard about some very interesting studies that are raising questions that will require further discussion. There is still much to learn from past studies and biological samples, but also study participants without whom the research would not have been possible. The Global HIV Vaccine Enterprise is keen to continue providing the space to allow for science to be presented, to be discussed, and also to be challenged. We had a peak attendance of 145 today, and that we could organize this event at such short notice is an indication that the HIV field can emulate the COVID-19 response in coming together quickly and moving fast. After all, in many cases, we're talking about the same people. Let's not forget that we are commemorating 40 years of HIV response next month, and we still don't have a safe and globally effective HIV vaccine. This discussion will be available online next week on the Enterprise website, www.vaccineenterprise.org. For now, it's thank you and goodbye.